Hi everyone. In this video on the plasma membrane, we're going to look at water balance, how it's maintained, and how it differs in plant and animal cells. Osmoregulation is the control of water balance, and it depends on the balance of solute concentrations inside and outside the cell. Osmoregulation differs between plant and animal cells. And before we get to look at the specifics, have a think and see if you can figure out what the structures are that cause the difference between the two cell types. We're going to be looking at how solutions of different tonicity affect the amount of water inside the cells. And these terms are always relative to the thing we're comparing, usually the solution outside a cell relative to the inside. If our solution is hypotonic, then it has a lower solute concentration than the inside of the cell. If the solution is isotonic, then the solute concentration is the same as that inside the cell. And if the solution is hypertonic, then the solute concentration is higher than that inside the cell. Let's have a look at how these different types of solutions affect animal and plant cells. We're going to start with animal cells, and for this example, we're going to consider a red blood cell. If we take our red blood cell and place it into a hypotonic solution, then water will move towards where the solute concentration is higher, which in this case is inside the cell. The cell will take on water, expanding until it lyses, rupturing the plasma membrane like an overinflated balloon. So what if we take that same cell and put it in an isotonic solution? Because the solute concentrations are equal, water uptake will be balanced by water loss, and there will be no net change in the volume of the cell. If you've ever been given IV fluids or have seen a medical TV show where they give someone fluids, you might have noticed that they give isotonic saline rather than water, and this is the reason for that. If you were given IV water, your plasma would become hypotonic to your cells, and your blood cells would explode. With isotonic saline, the concentrations in the plasma and the blood cells are balanced, so the volume of the cells remains stable, and this is the ideal situation for animal cells. What about if we put our cell into a hypertonic solution? Remember, this means that the solute concentration in the solution is higher than that inside the cell. If we put our cell into this beaker, water will move out of the cell towards the solutes, causing the cell to shrivel up in a process called crenation. We mentioned before that the effects of tonicity are different in plant and animal cells, and hopefully you figured out the structures responsible for this as being the cell wall and the central vacuole, neither of which are found in animal cells. Let's start by putting our plant into a hypotonic solution with a lower solute concentration than the cell. Like an animal cell, a plant cell in a hypotonic solution will take on water, but unlike an animal cell, the plant cell has a rigid wall that will prevent it from rupturing. The excess water will be stored in the central vacuole, which will then create turgor pressure outwards against the cell wall, giving the plant rigidity and strength, making this the ideal situation for plant cells. What do you think will happen to a plant cell in an isotonic solution? Well, much like the animal cell, not much. The plant cell won't take on any extra water. But unlike with animal cells, this isn't an ideal situation for plants. Without the turgor pressure from a full central vacuole, the plant will become flaccid and wilt. So, next time you water your plants, make sure you use water rather than isotonic saline. Neither plant nor animal cells are happy in hypertonic solutions. The plant cell can't crenate like the animal cell, again due to the rigid cell wall. But it can undergo a similar internal process called plasmolysis, where the plasma membrane pulls away from the cell wall due to excessive water loss. Thanks for watching, and if you haven't seen them yet, please make sure to check out the other videos in this section.